Welcome to the Applied Biosystems Taq Talk video series, where we break down real-time PCR to make it easier for you. Single nucleotide polymorphisms, or SNPs, are occurrences of a single nucleotide difference at a specific spot in a genome. If this variation occurs within a specific gene locus, then that gene is said to have multiple alleles. While most SNPs don't have a big effect, some of them correlate with disease, drug response, and other phenotypes, and those are of intense interest to researchers. Luckily, Applied Biosystems TaqMan SNP Genotyping Assays are sensitive, affordable, and easy-to-use method for differentiating between SNPs. Let's talk a little about what makes SNP assays unique and how you should go about analyzing your genotyping data. All Applied Biosystems TaqMan real-time PCR assays consist of target-specific primers and one or more probes optimized for specific types of measurements. Like a gene expression assay, a TaqMan SNP genotyping assay contains sequence-specific forward and reverse primers to amplify the polymorphic sequence of interest. However, SNP assays are unique since they contain not one, but two Applied Biosystems TaqMan MGB probes. One probe labeled with VIC dye detects the allele 1 sequence. Another probe labeled with FAM dye detects the allele 2 sequence. Note that all genotyping assays use the same notation for the context sequence, which is the immediate sequence around the SNP. The first base in brackets is detected by the VIC probe, and the second base by the FAM probe. You can find all this information online for every assay. The assays are combined with master mix and 1 to 20 nanograms of purified genomic DNA and then amplified. Data can be collected in real time or offline, and the analysis is taken from the endpoint read of the fluorescence. The sample will either be homozygous for allele 2, which means we will see mostly FAM dye, homozygous for allele 1, which means we will see mostly VIC dye, or heterozygous, which means we will be about equal contributions of signal from each dye. When viewed across an entire plate of samples, the data will typically resolve into 3 discrete clusters, assuming enough samples were used to display a normal distribution, given the minor allele frequency of your SNP. Note that there are times when you would not expect to see 3 clusters, such as when the gene is on the X chromosome, when a copy number variation is involved, or when the minor allele frequency is extremely low. When reviewing SNP genotyping data, it's critical that you remember that SNP assays are qualitative, not quantitative. Let's take a closer look at what's happening in each reaction. For example, this sample is a VIC dye homozygous call. However, if we look at the real-time data, we'll see some amplification of both probes. Wait, why is that? Well, the probes are specific for the respected targets, and most of the time, they bind to the correct allele. But depending on the surrounding sequence, there can be a low amount of mismatched binding from the opposite probe. This is normal, and one of the reasons why assays are a qualitative measure of the sample, not quantitative. The software makes calls based on the dye ratios and cluster orientation, and not the CT values. If we look at 2 respective samples, we can see there's a difference in the amount of VIC amplification. So, even though there may be some mismatch binding, we can still differentiate the clusters. To account for mismatch binding, a quality value, which is the probability of a genotype call, is assigned to each well. Most of our software sets the default value for this at 95%, but you can adjust it manually if desired. A data point with a quality value lower than the default threshold does not pass quality specifications and will be an undetermined call. By lowering the default quality threshold, you can change an undetermined call to a genotype call. Also, note that running a no template control sample is helpful for proper calls to be made. We recommend using our free Applied Biosystems TaqMan Genotype software with its advanced algorithm as the best program for making genotype calls to remove the guesswork from your analysis. For more information about SNP genotyping using real-time PCR, check out thermofisher.com/TaqMan SNP. Check out other Applied Biosystems Taq Talk videos on our website at thermofisher.com/taqtalk. And thanks for watching. See you next time.